This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 20, recorded December 15th, 2010. Hi, everybody. I'm Vincent Racaniello, and I'm with Professor Dickson Despommier.
Hello, Vince.
How are you, Dickson?
Feeling chipper.
I've spent the whole day with you.
You have.
Shall we discuss what we did?
Sure.
We're working on putting West Nile's story in ebook format.
Right.
We can put it on all these ebook stores.
That's right.
And it's a small book, so we're testing the waters,
We're
right?
practicing.
But first, you have to be accepted.
That's right. I have to jump through a few hoops.
Into the iBook store. But perhaps in a few weeks, we will have it up for sale, and that will be followed by...
Parasitic Diseases.
That would be cool.
5th edition.
And followed by other things as
Parasitic edition 6.
6, etc. So the ebook format can be a boon for academic tomes,
This
right?
will save a lot of trees. It will probably put traditional publishers out of business and create others.
Saving trees allows them to grow and regenerate oxygen.
And sequester carbon.
Yeah, which is what they're meant to do.
Absolutely, yes.
According to us. Well, in the whole scheme of the world, they help everyone, not just people.
Of course.
Make more oxygen.
And purify the air with their leaves.
And that, combined with vertical farming, will give us back the forests.
There you go, Vince.
Simple.
There you go.
Very simple.
Yep.
Well, this is This Week in Parasitism, TWiP. We are near the end of 2010. How about that, Dickson?
That's,
Did you think it would ever happen?
A life-defining moment.
Yes,
I did think it would happen, actually. I did.
You know what's quite interesting?
Go on.
You start a year,
Yeah,
and you say, wow, I got a whole year ahead of me.
that's right.
And before you know it, it's over.
You got it.
And such as this year, December 15th, and in a few days, you're going away for the rest of the year.
True.
And for a good part of next year as well.
10 days.
10 days.
10 days is not a good part
So
of
this
next
will
year,
be the ultimate TWiP of 2010.
It will.
Just let's do a review.
Sure.
I know, let's see when we started TWiP. We have to go
historical jaunt back in time.
We'll go to microbeworld.org/TWiP.
Okay.
It really comes off the tongue very nicely. And we will go to episode number 1.
Yep.
Let's see what the date on that was. I'll have to go to the last page. Episode 1. Do you know what the title of episode 1 was? Introduction to Parasitism, posted October 6th. No, that can't be. I was going to say 2010. That was only a few months ago.
2009.
Posted November 13th, 2009.
Okay, so we're more than a year old.
A little more than a year. 20 episodes.
We can promise more timely episodes from now on.
Okay. Is that our New Year's resolution?
That's our New Year's resolution. And to have a list of illustrious guests.
Yes, because we will go through all the basic stuff and then start to bring in some guests. Is that the plan?
Sure. You get the basics down first and then go back and learn what's being done on them. And in particular, things like malaria, leishmaniasis, hookworm. Those diseases all have big groups out there working away trying to find out their little secrets so that we can take advantage of them.
Very good.
So...
what brings us today? No, that's just totally wrong.
No.
What brings us today? Let me just tell you what we did last time.
Yes.
I have to go to the
We did the pinworms last time.
Echinacea preventius. Sorry. Enterobius vermicularis.
That's right.
The pinworm.
The pinworm. And surprisingly, for such a benign little organism, we spent an inordinate amount of time on it.
But it was really good.
Yeah, because it laid down the principles for all these worm infections that we're going to talk about now.
We're going to... Right, so that was a worm. It was a nematode.
It was a nematode.
So that would put it in the same chapter as today's subject.
This is correct. There is a theme to this. which we will continue into the new
Hey,
year.
look at this, the nematodes. In your book,
Go on.
Enterobius vermicularis.
Yeah.
The next one, Trichuris trichiura.
My goodness.
Are we going to talk about that today?
We are.
And I think Ascaris lumbricoides,
Correct.
which is
The giant intestinal worm.
not a hookworm.
No.
That's next.
Yep.
I don't know if we'll get to both of these because the way we go.
That's right. It becomes a conversation rather than a presentation, which is the best
But
way
let's
to do
start
this.
and see what happens with Trichuris trichuria.
Or trichiura.
Trichiura.
Trichiura. So it's got a common name. Almost all these do. The pinworm, Enterobius vermicularis, is the common name, is the pinworm. And this worm has a common name also, and it's based on its shape, as well as the pinworm's a common name was based on its shape. This worm, when you look at it in one way, of course, if you look at it in another way, it's a nematode. But it's an unusually shaped nematode because one part of it is long and sinuous, and the other part is kind of thick and less sinuous. And
Yes.
when you look at it from the hind end, which is the thicker end of the two ends,
For both sexes.
for both males and females, it has the general characteristic of a whip.
A whip.
It's got the handle of the whip is the rear end of the worm, and then the anterior portion, which is much longer than the posterior portion, that part of it is the whip-like area.
I don't know, Dickson. I think you need an imagination to think it's a whip.
Well, you do, but I mean, no. Look at the one at the top. Look at the figure number 17.1. That's a female Trichuris, and to me that looks like a whip.
Okay. Well, we won't debate that because it's entrenched.
Correct.
But
the point is that's what it got stuck with in terms of its common name.
Now,
and
in
it's
your book here, sorry,
it's
go
quite
ahead.
big, by the way. This is a big worm.
How
This is
big
not
is
a
this?
small worm. Well, it's centimeters long.
As opposed to
The
Enterobius.
pinworm was millimeters long, and this is centimeters long. You can see this darn thing with your naked eye. This is an easy one to see.
And this lives in your gut.
It lives in the large intestine only. And it lives in a very special niche. Pinworm, remember, was sort of free-floating and swam, so to speak. It's kind of moving back and forth against the flow of feces that continues to move out, right? But not in a continuous fashion unless you've got diarrhea. This parasite lives embedded. The interior portion of this worm is woven back and forth and back and forth and back and forth into a row of columnar cells in the large intestine. And it has the ability to cause the columnar cells to actually fuse together to form a syncytium. So my term for this is an intramulticellular niche. This worm lives as an intramulticellular parasite for its anterior end.
The anterior is the thin end?
That's correct. The head end is the thin end. So the tail of this worm actually sticks out into the lumen of the large intestine.
This is the broader part.
That's correct. And that facilitates the expulsion of eggs.
So the eggs come out of the larger part?
There's a little pore somewhere
Yeah,
along the worm's length, which is not included in the anterior part of this worm, which allows the eggs to actually exit into the lumen of the large intestine.
So it looks like the female has more whip-like area, the wider area than the male.
That's true.
Is that because she has to make eggs?
This is exactly right. She's an egg factory, basically.
So these guys reproduce sexually.
They do.
Always. No asexual stage.
Nope. They're sexual organisms from start to finish, just like pinworm. Pinworm is sexual as well.
And the head, the anterior part, which looks very pointed here.
Yeah.
Is there any differentiation about that?
There's a tremendous amount of structure.
Is there a
there
nose?
is. There's
eyes.
No eyes. Nematodes don't have eyes. So that we can stop at that part. But it has a brain. Okay? It has a ganglion that is a collection of neurons that coordinates the movement of this worm. And all kinds of movement occurs here, including the fact that it has to move eggs along the oviduct before they're actually expelled.
Does it have a mouth, did you say?
It does have a mouth.
Is it taking food via the mouth?
Well, Vince, that's the curious part of this little worm.
It just talks with its mouth.
It doesn't even do that. Remember, it's embedded in a syncytium of large intestine columnar cells, right?
Mm-hmm.
Now, if I take you back to our description of Trichinella, we had some iPodcasts on that as well. We talked about the adult female worms and male worms, as they are, too, embedded into the row of columnar cells in the small intestine. It turns out that Trichuris and Trichinella are related to each other. Okay? The order that they're part of is called Trichiura.
An order.
An order.
What's an order, Dick?
An order. Let's see. If you're in a restaurant, of course, it would be, I'll have the entree. No. Okay. So an order is a group of organisms sharing common features. I'm not about to pretend that I'm a systemicist, but if we start with the concept of phylum, and then class.
Kingdom, phylum, order.
Kingdom, well, okay, you start with animal kingdom. Or
Kingdom, phylum, order, class, genus, species.
That's right. So the order,
variety,
the
blah,
order
blah, blah.
is just a little bit higher up in terms of hierarchy than family.
Yeah, order is above family.
That's right.
Phylum, class, order, family. We forgot the family there.
Yeah, we did.
Kingdom, phylum, class, order, family, genus, species, variety, race.
There you go.
I remembered from college.
You were pretty smart in college,
No,
too.
I wasn't smart.
We're not going to go into that, Vince, but I know better. So the point is that this worm is related to Trichinella in many ways, including the fact that it contains a row of cells which starts very near the head end of this parasite and continues all the way to the junction of the thicker part of the worm.
Is there a name for the thin part versus the thicker part? Yeah, thin versus thin, Vince. I'm just asking.
No, we don't do that. It's the anterior portion of the worm.
So the anterior is considered the thin part.
It is the thin part.
And then the posterior, is that what we would call it?
We would.
Is the thicker part.
That's correct. So, but the point is that there's this single row of cells that lines the esophagus of this worm all the way down to the junction.
Hey, that's like the single row of cells in Trichinella, which do different things, right?
That's exactly
Remember
correct.
that. What are those called again?
Stichocytes.
Stichocytes.
And the Greek word for row of cells is stichocyte. So it's a Greek word that says the same thing as an English word, except that you're using Greek. So, but
Nice.
because we don't understand the function of those cells. Mm-hmm. What we don't also know is whether these stichocyte cells perform similar functions to the ones that Trichinella has. The assumption is because they live in similar niches in different places throughout the small intestine and large intestine, but they're both occupying intra-multicellular niches, that the same functions occur in both Trichinella and Trichuris. Mm-hmm. There are some other worms that are part of this group. One of them is called Capillaria, another is called Trichosomoides, and they all seem to prefer to live inside cells as adults. Mm-hmm. Trichosomoides actually lives in bladder cells, the squamous cells that are lining the surface of the bladder, which is an unusual place for any parasite to locate.
This is a human parasite?
Trichosomoides. No, it's an animal parasite. But Capillaria can be a human parasite, or a parasite of humans.
This reminds me of that little fish thing that goes into your bladder. What was that again?
Yeah, that was called Kanduru.
That's not a parasite, is it?
It is. Of course it's a parasite.
It's a big parasite.
Yeah, because it swims into the gills of these large catfish in the Amazon, and it actually rakes across the capillaries of the gills, and it sucks blood. So it's a blood-sucking organism, like a leech. All right? You might want to look at them as predators, but predators are usually larger than what they feed on. In this case, they're parasites.
So the Trichinellas are in the same order,
but
they have different families, obviously.
That's right.
Trichinella is the family. No, Trichinella is the genus. What's the family for Trichinella?
Trichinellidae.
Trichinellidae.
Yep.
And for Trichuris?
I guess it's Trichuridae.
Trichuridae.
Sure.
Is Ascaris is also a different family?
Much different.
Okay.
Much different.
So why were these first discovered? Do they cause disease?
Why or how?
We could do both.
Well, I'm sure there were incidental findings on autopsies in the very beginning of the field of parasitology, because in those days we didn't associate diseases with entities very well. It took a long time, in fact, to even associate disease with Trichinella, all right, back in the 1800s. And this parasite was known about before that, because other animal species can also acquire similar parasites. And We'll see what the significance of that is later, Vince. But there's an interesting connection that's
later
emerged.
today or later in the
I
series?
hope later today, unless I get tongue-tied and can't finish this presentation.
So this is called a soil-transmitted helminth.
It is. It's a geohelminth.
Is that the case for... No. Last
Pinworm?
week's pinworms, no.
Nope. It never does make it to the soil, actually. But it could be airborne, for
Right.
instance. And it could be dustborne. You could imagine
Yeah,
it.
not soil, though.
But they dry out so fast and die that it's probable that the main transmission mechanism is through the fingernails.
And it says here it's one of 3 major soil-transmitted helminths.
That's correct. The other 2 being ascaris
Mhm.
and hookworm.
Okay.
And there's a 4th also. That's strongyloides, which we will talk about as well.
All right.
So back to Trichuris. So
Yes.
Trichuris, there is a variety of species of Trichuris. There's one that lives in sheep. That's Ovis. There's one that lives in dogs. There's one that lives in mice.
That would be Canis, the dog one.
Yep. And
Mice
the one that lives
would
in mice
be-
and Murus.
Murus.
That's right. This one lives in pigs.
And do they cross species?
No, they do not. Suis is the one that lives in pig.
Mhm.
So all
Cows, bovis.
don't know, Vince. Don't know.
All right. And then human is trichuria.
Trichiura.
Trichiura.
Trichuris.
That's tricky, Dickson.
And you know what? All the students have trouble with it too.
Trichiura.
Trichiura.
What does the name mean? Trichuris trichiura.
Well, the word trick
Yes.
is a Latin word that refers to hair-like.
Ah,
So trichinella is a little hair. So Trichuris.
and Racaniello is a little rock.
Okay.
Sorry.
It's okay.
Trichuris, they both have trick in the name. Trichuris,
They do.
the genus species,
And
trichiura.
If you look at them, you'll understand why they named them that, because they're hair-like.
So the trichiura does not have a human specification, as does the
No.
ovis and
No.
the murus, etc.
No, Trichiura is not a...
Was this the first one discovered, the human?
I don't think so. I'm pretty sure that they looked in other animals like pigs first and maybe rats.
In your book, it says it was discovered by Linnaeus in 1771. Now, wasn't he involved in classification?
No, he didn't discover it. He named it.
He named it.
All of those little abbreviations are the names underneath these words of parasites. Those are the guys who named the
them.
parasite. But he didn't discover the parasite.
He classified it as a nematode in
That's right.
1771.
Sure.
Who discovered it? Do we know?
We do. Don't we say there?
Well, it says it was described by Morgagni in the Secum,
There you go.
but that was 1740.
Okay.
Ah, that's earlier than 1771. Yes. My God.
I don't commit that kind of knowledge to my
No,
memory.
that's not important. But I'd like
That's
to
why
give
we have books.
I like to give a time frame to the discovery.
Yeah.
So this is
So it was the age
Early.
of discovery of infectious agents.
very early.
Yeah,
Because remember, the first vaccines were in the late 1700s.
Yeah, but that's later on. Look at tapeworms. Tapeworms were discovered early because they were big and easy to see.
Sure.
Ascaris was certainly around for a long time, known about at least, but never known with regards to its life cycle or whether it had a life cycle or not. So a lot of this early work was sort of shooting in the dark, so to speak. We found this, we found that, we found this. A lot of description without explanation.
Dixon, it says here that they have been these trichuri have been identified in
Coprolites.
coprolites,
That's
which
correct.
are petrified.
I knew
feces,
what you were going towards there.
petrified feces of prehistoric humans.
Correct.
That's probably the case for a number of
Yeah.
these things. So what was found, the
eggs,
which must be very stable.
or the shell of the egg is very stable, and you can do a microscopic examination and find out who had what. We're going to get to this life cycle, and once you see how it's transmitted, you'll understand why virtually every single person living on Earth, probably of 200,000 years of history, all of us probably had these.
Are you serious? So this is even more successful than toxoplasma?
Well, gee, you're going to pin me down on this one.
Well, you said toxo was like the most successful, but
it's because it infects all animals, Vince.
all animals, yes.
Toxo infects all tissues in all animals.
Yeah, but this doesn't infect whole
No,
animals.
this only infects
But humans it infects, and it's very good at infecting all of us.
It's extremely good at it.
But many of these parasites seem to be good at that.
They are. That's why they're still here.
Hmm.
In fact, it's a normal occurrence to have parasites. It's an abnormal occurrence not to. And we'll see the consequence of not having this particular parasite.
Would that be because we're a successful system?
We are an enormously successful system.
Now, so we have parasites.
We have parasites, but we've also learned how to get rid of these parasites. And in doing that, we have created other problems for ourselves.
Now, which one was it when the animal dies, the parasites pass to someone else because another animal eats it, right?
Trichinello.
Trichinello.
Very and
smart.
toxoplasma as well.
Smart.
You betcha. Well, I wouldn't claim they're smart.
Well, evolutionarily smart.
They were selected for
exactly.
life. And that's a great mechanism for transmission, right? Okay, so let's find out how this thing is transmitted, right? They're big worms. They live in the large intestine. You asked me whether they cause disease or not, and the answer is yes, they do. And in fact, this could be a fatal infection in some cases.
Mm-hmm.
The major results of infection with this are anemia, right? And they could even induce a form of malnutrition because of the diarrheal disease associated with it, and they can also induce something called malabsorption syndrome.
But they don't always do that, it sounds.
They don't. It's a dose-dependent thing. So
how many worms you got?
Yeah, a couple of worms, nothing. A lot of worms, something, and a whole bunch of worms. And it depends on the person and how old they are and their other nutritional status.
Okay.
Okay?
So how do we acquire this?
All you have to do, Vince, is ingest a little bit of feces along with your meal.
Which I understand is not difficult to do.
Not at all. In fact, I know that sounds disgusting, but probably we've all done it.
Really?
Because of food handlers.
Food handlers, sure. That's how we get norovirus infections.
But you know what? If a food handler had this infection and they contaminated themselves, let's say after they went to the John and says all employees must wash hands, it doesn't say when, especially if you don't speak.
Before leaving.
You don't speak more. If you don't speak English, you certainly wouldn't obey that sign. But the point is that this egg has something special about it that. Unlike pinworm, which is almost immediately infectious after it comes out onto the oxygenated environment, this egg takes about a week to embryonate.
So the egg is present in soil.
Yes, it takes about a week.
It's been deposited there with feces, fecal contamination.
That's right.
And it's very stable.
It's extremely stable. In fact, in sludge, in treated sludge, in a layer that the Germans referred to as the Schmutzdecke, which all microbiologists know about. It's the anaerobic layer of a digestion plant, of a water treatment plant, rather. The sludge that collects there, if it contains these eggs, these eggs can remain viable for years.
Wow.
Years,
But Vince. They do not embryonate until it's a more favorable environment?
No, they can embryonate and stay in a resting stage as a larva inside the egg. In fact, this egg is not infectious until it does develop to a larva. So if you were to have food contaminated with eggs from a food worker in a restaurant, say, for instance, a maitre d' or a waiter that comes over to your table and mixes the salad in front of you with their hands, of course, as they've just come back from the John, of course.
Or they could do it in the kitchen. You wouldn't.
A lot of them wear gloves now, probably to protect themselves rather than you. So they're mixing the salad, and if they were to be passing eggs from Trichuris, and that got into your salad, and you ate it, those eggs would go right on through. They wouldn't hatch.
Really?
That's correct.
So they have to embryonate they have to hatch in the soil?
Outdoors. They don't hatch in the soil.
They
embryonate, and then they stay that way.
What does this mean to embryonate?
The best example I can give you, Vince, is to imagine this as a chicken. Okay. Everybody understands chickens. When you go to the store and you buy chicken eggs, they're unembryonated.
Right.
Correct? In fact, most of them are sterile.
You have yolk and...
Nothing.
Yolk and...
albumin.
Well, that's one of the proteins. It's a major protein.
Well, it's called albumin. No, that's not the right albumin. That's referred to as the albumin, right? And you can coagulate it with high temperatures, and you've got a hard egg.
It becomes white when you cook it.
Yeah.
What is that called?
Albumin.
It's just albumin.
The white of the egg, Vince.
Actually, we grow influenza viruses in embryonated eggs.
Ah, that's different.
It's grown in the white stuff, which is chorioallantoic fluid.
Yeah, but that's different. So if you can imagine...
Yeah, because there's an embryo now, so it's different.
That's right. Yeah. So that's the deal. I mean, eggs are eggs.
So when it starts to develop into an embryo, that's embryogenesis.
Embryogenesis. It takes about a week.
And that's what these guys are doing. So in the picture, you could see the difference. The embryonated, it looks like a worm.
It is a worm.
It's still inside this shell.
Remember how long it took pinworm to embryonate? 6 hours. That's remarkable, Vince. Same number of cells.
Days.
Same number of cells.
Now, why is it different?
I don't think I can answer any questions
It's
that
just
begin
the
with
way
why.
evolution has selected.
Yeah. So pinworms are immediately infectious for the next host, whereas this thing has to wait in the soil for a week, but it can wait for years afterwards.
Now, wait a minute. I don't quite understand. It can wait before it embryonates, you mean?
It can wait to be ingested by a host.
So if you eat an embryonated egg, then you'll be infected. But if it's not embryonated, it passes through.
You got it.
So it's a matter of luck.
Yeah.
How long this egg has been sitting before you eat it.
This is very true.
So typically you're getting it from food handlers?
No.
No.
You're not. You're getting it from random acts of defecation in the environment from little kids who have this infection, who perhaps suffer from loose stools or diarrheal disease, and who are allowed to defecate ad libitum, as they would say. Or to populations that haven't got the privilege of sanitation.
Good sanitation.
That's right.
But in our country, for example, daycare centers could pass it, right?
Not really.
Because it doesn't have time to embryonate?
That's correct.
Ah.
Now you got it.
But the food handler...
Yes.
If the food handler was just in the bathroom and contaminated his or her hands with feces, probably didn't embryonate.
It takes a week.
And it has to be out of the host to embryonate.
It takes a long time. And the next one we'll discuss, Ascaris, is exactly the same as Trichuris.
I'm just trying to determine what conditions would allow for it to have a week somewhere, not inside the intestine.
Ad libitum diarrheal disease of children in rural settings.
So it goes out. But the diarrhea...
fecal events can occur anywhere in the outdoor environment.
And you're saying that there's contamination remaining.
Yes.
You can't clean it all up.
No. You don't clean it all up. The kids go into the soil. They go behind a tree, or they just do it on the sidewalk. If they do it on the sidewalk, of course, they die.
But peri-domestic, or in the house.
Peri-domestic.
So in the house, it's not likely to be transmitted.
You'd clean that up, or you'd have diapers on the kids. I think good sanitary practices would prevent this from spreading in a normal environment.
Okay.
But that's not the case in most places. It's obvious it's not, because you know how many people are infected with this?
I don't have a clue.
Hundreds of millions of people.
That's nothing. There are billions of people on the planet.
Well, we might have 1 billion people.
What's the population of the planet?
6.9 billion.
7 billion.
You might have a third of the population of the world.
Infected? Are you and I infected?
With one of these three things.
No, likely.
But Harold W. Brown, who was my mentor, and the reason why I became a parasitologist to begin with, was infected with Trichuris. And when he had his appendix out, they found a Trichuris living inside the appendix.
So they hadn't known he was infected until he got inflamed. Was the inflammation of the appendix due to the Trichiura?
We don't think so.
Trichuris.
We don't think so.
So if he hadn't had an appendicitis, he probably never would have discovered this. So there are many asymptomatic infections.
Tons of them. Remember, it's dose dependent.
So I could have it.
Nah, chances are that you wouldn't never even encounter this. Where would you encounter it?
A food handler.
I told you those are not going to give it to you.
Without doubt.
A food handler will never give you this infection because the eggs have to sit in the ground for a week.
In the ground,
Let's
nowhere
review.
In the ground, nowhere else.
Yeah, that's right. It has to sit outside.
Could it sit on my desk
It'll
here?
dry out and die.
Soil is the only place?
Well,
a lake? Schmutzdecke?
Schmutzdecke would be good.
A tree hole?
Tree holes would be fine.
All right, some moist environment.
Usually defecate inside tree holes.
All right, now I'm starting to get it.
Good. Don't get it.
I told you I wasn't smart, Dickson.
Don't get it. No, you're just persistent in your ideas.
How many infections are there in the U.S., for example?
We don't know.
You said in the world there's hundreds of millions.
Well, it might even be over 1 billion.
But there probably are some in the U.S., right?
There are some. Of course there are some.
Now, when you were a parasitologist, a technician, I know you.
Yeah.
I didn't mean to insult you, but
No,
I
that's
meant
quite right.
as a technician with Dr. Brown,
did
you ever see Trichuris infections?
Of it. Yeah, we had lots of it. You know why? Because we had a big immigration from Puerto Rico at that time.
Where it's endemic?
A rural migration of people from the surrounding countryside in Puerto Rico.
Later on, you'll tell us how it's diagnosed.
Yeah, it's pretty easy, actually. You just look under the microscope.
At what?
And find the eggs, feces.
Because for some, you don't find them in feces.
That's right. Remember which one?
Pinworm.
There you go.
You have to put a piece of tape.
You see, you are smart.
No. Piece of tape, right?
Yeah. Piece of clear sticky tape. For this parasite, however, she produces this female worm after they mate.
Where do they mate in the intestine?
You know, we don't have a clue. I'm going to be honest with you here, Vince. There's a lot of missing information here.
But these guys don't exist anywhere else except as eggs. Outside of the host,
They won't. No, the mating has to occur within the host, but nobody knows where, and nobody knows when.
At night, Dickson.
It's always nighttime down there. These worms live in a very special milieu, and what goes on in that milieu is anybody's guess.
Look, if it's the only place where the worms mature, the large intestine, then they must mate there.
They must mate there, but
we just haven't seen it.
Or maybe on the way down to the large intestine, because these worms start out really small.
So we've missed something here.
Have we? No, I haven't finished this yet.
You ingest an embryonated egg.
And then what?
What happens next?
It then passes through the esophagus and stomach, down through the small
intestine. It survives
intestines.
the stomach and the jejunum, so it's still an egg.
And it must receive an acid environment cue first, and then a basic environment cue next, and then a third cue. Okay, now here's the intriguing part. This is very intriguing. This is a recently discovered thing, like maybe a month old.
Wow.
I know. So I get to tell your listeners and my listeners, I should say our listeners, something that we couldn't have put into this book because we didn't know it. Okay? How does this parasite know where it is? How does it?
GPS.
Gut positioning systems. Maybe that's true, but I must tell you something. Here's the clue to this one, is that it's very difficult or impossible to infect germ-free animals.
Oh, so it tells where it is by the microbiota that it encounters.
You'd think so, but that actually...
So germ-free mice cannot be infected?
Cannot be. Well, I didn't say can't be, but it's very, very, very difficult to infect them. And
curious.
So what they found out by investigating why that is that E. coli turns out to be an essential ingredient in its attachment mechanism to the gut tract.
But there are many other...
So remember, viruses have to
attack.
They have to attach first before they actually infect. This egg turns out to have to attach first in order to infect. And so how does it attach? It doesn't do it directly, because if it did that, it would have attached to germ-free animals, right?
Right.
What they found out was that E. coli forms a bridge with its pili between the surface of the egg and the surface of the large intestine.
Wow.
Wow is right, Vince, because for years we labored under the impression that this egg hatched in the large intestine because it received an environmental cue that was there. But that obviously is not what happens. What happens is the egg encounters something like Velcro, right?
It's like bacteria, right?
Yeah, it's the double bond between the gut tract and itself, and it stops the migration of the egg, and that's when it gets its cue to hatch.
So
not before.
Is it just E. coli, or can it be other bacteria?
Well, I don't know the full story on this. It's emerging as we speak. They're still investigating other species of bacteria as to whether they can substitute for E. coli, but certainly E. coli itself can do it. And they know that it's a combination of the surface of the egg and the pili, the chemistry of the pili, which allows this to happen. So it's a non-specific attachment process, but it needs to be attached in order to infect. And that's what triggers the egg to hatch. I guess it receives cues from the large intestine. It's rather anoxic in its behavior. By the way, this worm is probably a microaerophil or an anaerobe. And the same is true for Trichinella, but not for the adult of Trichinella. It's only true for the larva of Trichinella, whereas in this case, it's probably true for the adult. Remember, the adult of Trichinella lives in the small intestine.
Right.
The adult of Trichuris lives in the large intestine. So, okay, so now
these have to have a bacterium to attach to the large intestine.
That is the going hypothesis based on the evidence so far.
Now, according to your diagram here, it says that the larvae hatch in the small intestine.
That was what the common feeling was about this.
So it's not right.
It's not right.
So they go to the large intestine, attach, and then they hatch. And this requires bacteria to bridge them.
So should we revise this textbook, that figure at that point would have to be drawn again to bring it up to date.
Yeah.
So now we're pretty current in terms of that. We don't know what the mechanism is, the exact chemical attachment process, but we do know that it's essential.
So the hatching means the embryo gets out of the shell
Correct.
and then it attaches in
So
this.
in order to get out of the shell, it has to digest its way out. All right? And the next parasite we'll discuss has the same problem. And the way it gets out is to elicit a protease inside the worm itself by the environmental cues that tells it now's the time to hatch. And it synthesizes the protease, it digests the eggshell, and it immediately burrows into the columnar cells of the large intestine. And it's a teeny tiny little bitty worm that's only about 90 micrometers long and about 7 micrometers wide.
Right.
3 months later, Vince.
It's burrowed in their anterior portion.
3 months?
I presume the anterior portion is burrowed into the colon, right?
It is. And in fact, if we go back to the other page for a moment, the other page in the other direction, you will see a scanning electron micrograph picture of Trichuris murus. And what you're looking at is the exteriorized posterior portion of the worm with a few eggs outside, and the head of this worm is sinuously wound back and forth among the columnar cells, forming a network of syncytium of the host and the parasite. It's an intimate cellular relationship. Now, there's another interesting structure on this worm, which I don't think I even mentioned it when I was talking about Tracaniello, but I might have. It's the hypodermal gland cells. It's 2 rows of cells, one on either side of the cuticle. Okay, so this worm has a cuticle, all right, which has got a very complex structure to it. And there are 2 cells per, if this were a segmented worm, you'd say 2 cells per segment, but it's not a segmented worm, but they're lined up symmetrically anyway, as if this worm were at one point segmented, but it's not. All the way down the anterior portion of the parasite. It's not on the posterior portion. It's just on the anterior portion. There's a plug when you look at the ultrastructure of this thing, there's a plug of material and a break in the actual cuticle itself. The cuticle actually folds inside and forms this rugated, membranous pocket that gets filled with stuff. Nobody knows what this cell does, but there are a lot of them in this parasite. And one of the speculatory themes revolves around the need of this parasite to regulate its osmolality. And so this might be a primitive way of allowing fluid to equilibrate with the extracellular matrix of the syncytium that the parasite induces. Now, I hope everybody that's listening to this says, this is not a simple worm. I hate to tell you. This is a simple worm compared to a lot of others. And yet it's still impossible to describe in full the biochemical mechanisms, the physiological needs, the nutritional status of the parasite, and the pathological effects of this parasite on the host. We don't know anything about a lot of this, okay? We just know that this is a bad player in terms of malnutrition in kids. It elicits diarrheal disease and malabsorption syndrome and anemia. But the anemia is not induced by the fact that this parasite is capable or incapable of sucking blood. So I actually worked as a technician here for a while, Vince, back in 1962. And in 63, I was a graduate student. But in 1962, I worked for a wonderful woman. Her name was Dr. Eileen Pike.
This is not part of Dr. Brown's?
Yes, she was a PhD student in his laboratory. And her thesis problem was to investigate the possibility that Trichuris murus sucks blood or doesn't. So how would you go about investigating this? In those days, we didn't have too many tools. And so what she was trying to do was to inject the mice first before infecting them with chromium-51. Chromium-51 is taken up by red cells, and it's included into the cytoplasm of the red cell. And then if red cells are ingested, and then you get an anemia effect by ingestion of red cells, you could actually detect in the gut tract of this worm the chromium-51 and the breakdown of the red cells, okay? That's what her hope was, is to show how this parasite actually sucks blood from a mouse. Well, it turns out that mice don't get anemic to begin with, so that's too bad. But even worse than that is that no matter how hard she looked for the breakdown of red cells inside the gut tract of this worm, she could not demonstrate it. So using chromium-51 as the marker for red cell breakdown, this worm still looked as though it had the capacity, at least, of inducing an anemia without the direct ingestion of blood. And so there's no bleeding in the large intestine as a result of infection with this. There's
but humans get anemic, right?
They do. That's the point. And the stool sample is not guaiac positive.
It says here that there's some blood in the stool.
Only
Trichuris dysentery in children.
Well, dysentery, yes. Dysentery is different than diarrhea.
It's bloody diarrhea.
That's right. That's right. But that's probably as a result of straining, thinking you've got something to move out of your bowel, and it's actually the worms themselves, and you see how embedded they are.
So there's a big mass, is that it?
Yeah, and it gives the patient the feeling that they have to move their bowels, and they continue to do this until the rectum actually prolapses.
Really?
In many cases, it's a prolapse. That's the picture you're looking at.
So they keep thinking they have to evacuate?
Imagine thinking you've got this enormous amount of bowel material in your large intestine, and you strain like heck to get it to go out, and it's all due to the worms themselves.
Wow.
So eventually they do start to bleed, all right? But that's not the mechanism of anemia. We think it's a central bone marrow failure.
Wow. How
Central
do they do that?
bone, that's...
It's pretty far away from the colon.
It's in... It's as far away as you can get.
They could send chemicals in there, but
they could, of course they can.
But it's not a matter of these worms piercing capillaries and sucking blood out, right?
Nope, not at all.
Although you could imagine with a big enough load of worms, it could make you anemic, right?
And that's exactly it. I mean, you do get anemic with a lot of worms, but the mechanism of anemia is not from blood sucking. So even that remains a mystery. So the biggest point of the conversation here should be from the perspective of the worm. Mm-hmm. What does this worm need from us?
Right. What makes it grow for those 3
Why
months?
is it a parasite?
What does it need? We don't know.
We haven't got a clue.
Clearly it needs something.
Well,
and it grows in the course of those months, right?
It needs us.
In the 3 months, it becomes a full-grown adult?
That's right.
All right.
So that's called the pre-patent period. Pre-patent period. The period before you can detect the parasite in the stool.
So after that period, they start to come out into the stool?
No, the eggs start to be
produced. The eggs come out. So how does that happen?
Oh, they just get extruded through the birth pore over here.
And then they're fertilized externally?
No, they were fertilized prior to that.
So they mate?
They mate. The female stores the sperm, and as she makes eggs, she fertilizes them as they come out.
And they start to come out after 3 months. Okay.
Yeah, and Trachanella works the same way, by the way. So that's why these two groups are connected together in terms of that biology as well.
So you have to ingest more than one egg in order to make more eggs.
Vince, you got it.
Right, because if you had one egg that grew into a male or female, that'd be the end of the story.
That is correct. And big deal. If you have one of these worms, who cares? Harold Brown had one worm, and he didn't even know he had the worm, right?
Just one, really?
One.
In his appendix?
Yep.
Interesting.
A single worm.
You would never detect one.
You couldn't detect it on a stool exam unless it was a female, and she was producing sterile Trichuris
so that can happen. They can produce eggs without being fertilized.
Yes, as we'll see in the next parasite with Ascaris.
Interesting.
So, yeah, a lot of missing data here. A lot of missing data for a very common apparent.
So you start to make eggs, and
how many eggs, Vince?
So let's say you've ingested a couple of worms and they can reproduce. Do you ever get more worms made in you as a consequence?
Good question. And everyone thinks that just like bacteria or viruses or protozoans, which if you get one, you can have many more after that.
Yeah.
With nematodes, with one exception, and we'll get to that when we talk about hookworm and strongyloides, with one exception, one egg, one parasite.
Okay.
That's it. Because that's the juvenile stage of that parasite.
So I presume you could take in a lot of eggs, depending on how contaminated.
Yeah, again and again and again. You see, and
then
little kids can build up big levels. But in the meantime, of course, the body is reacting to this stuff and saying, wait a minute here. Something's going on.
Right.
That's not us. Okay. IgE, over here. IgA, over here. Eosinophils, attack. And it's what they do, and they attack this worm. Right. How can those little things affect the outcome of an infection with an animal that big? And they do. They make life very uncomfortable for this parasite, and so it withdraws and passes out as the result of immunity.
Is that right?
That is exactly right.
So it's self-limiting, you're saying?
It is.
In all cases?
Well, unless you're immunocompromised.
Okay. So how long does this process take?
Years.
3
to 5.
Why does it take years?
It sounds like a sentence for some guy who just held up a gas station. 3 to 5 for Louis over here. How long have we got Trichuris in us? 3 to 5 years. So every single day, this parasite can make 3,000 to 5,000 eggs for 3 to 5 years. Add it up. That's a lot of eggs. Have we got a calculator someplace around here?
Yeah, I do have a calculator.
Good. Let's do the math.
What do you want to know?
Per female that lives 4 years and produces 5,000 eggs per day, how many eggs altogether does she produce?
So 4 years, then we're going to multiply that times 365 days.
Yeah.
No, I just made a mistake. Let me do it again. 4
okay.
times 365.
Listeners that send this answer in before Vince will win a prize.
1,460 days times 4,000. 5,000 eggs?
Make it 5.
Would you like 6?
No, 5 is fine.
5,000 a day is 7,300,000 eggs over that 4-year period.
Right. And let's say that this has a 30% survival rate in the soil for 5 years.
That's pretty good. Still millions of eggs.
It's millions and millions of eggs. Now, Vince, when feces is deposited on the ground, what happens
This is only one female that you
That's
did.
one single female. Now,
it
what if you
says
had...
here that some kids can have worms the whole length of the colon.
That's right. They can have hundreds, sometimes thousands.
How much does it weigh?
What, a worm?
No, thousands of worms.
Oh, that would weigh something.
A pound?
I don't know. I mean, no one's ever weighed these
Let's go find
things.
out.
Actually, if you do this for Ascaris, you can find out, and we'll get to that in a minute.
We may not.
We might not. We might just spend this whole time on Trichuris because it's so interesting. But let me just tell you something about the ground and the ecology of the ground and why Darwin's first paper... What was his first paper on, Vince? Do you know?
I don't know, Dickson.
It was on the earthworm.
Wow.
Lumbricus terrestrius.
Mm-hmm.
Now, Lumbricus terrestrius is this, we would call them night crawlers, okay?
Big guys, yeah.
Right. So what is their job? Their job is to dig tunnels throughout the soil, eat bacteria and other microbial life, fungi too, and defecate the byproducts of their metabolism at the back end, which serves as a fertilizer.
Mm-hmm.
So composting, everybody knows about that. You take vegetable matter and just throw it into a barrel and buy the first load of worms and throw them in the barrel. And if you wait a couple of weeks, you look around, all those leafy green vegetables have been turned into soil. Then you just keep throwing your vegetable stuff in there, and the next thing you know, you've got worms coming out your ears. In soil, earthworms can travel long distances, and they come up outside, and they crawl around at night, because that's why they're called night crawlers. And when they encounter a large amount of bacteria, they just stay there, and they eat, and they eat, and they eat. In the same vein, no pun intended, during the daytime, birds, and you know the earliest birds catch those worms.
Why do the worms come out at night?
They come out to mate, to get oxygen.
To avoid birds.
To pick up the moisture on the grass, because they're slimy little critters. They have a whole bunch of reasons for coming out. And, of course, feces is referred to as night soil. So we have birds with their feet on the ground tracking across the environment. We have worms taking things from the ground and moving them inside the soil and then moving them from place to place. Pretty soon, if you took a time-lapse photograph of a pile of feces that someone had produced that day, just one day's worth, how long would it be before you couldn't recognize the site and it looked like the earth again? And the answer is not too far, not too much, okay? Maybe 2 or 3 days, maybe 4 days, and it's gone. What happened to it? It didn't evaporate. It was eaten and processed. And so Trichuris eggs that are deposited here
They get spread all over.
can end up, 15 yards over there someplace.
So the earthworms do that.
Right.
Because they pick them up and redeposit them.
They do.
The
they
birds
do.
eat the earthworms, and they defecate and spread the trichuris. And I
Vince,
presume that
you've got a medical ecology wheel turning in your head right
the
now.
trichuris eggs will survive the earthworm and the bird.
No problem
right?
there. None.
And some birds can go long distances, as you know.
That's right. So this parasite becomes ubiquitously distributed throughout the environment.
So if I went outside here in New York City and took a soil sample, would I find these?
Well, it depends on the species that you're looking for. There is one in dogs. Okay, I guess that's called Trichuris vulpes.
Dickson, if you just took dirt and sequenced it, what would you find?
Ooh,
a lot of stuff. Right?
Oh, Vince, you might discover the next antibiotic.
Well, I mean, it sounds to me that there's we know there are a lot of bacteria and phages. And now it's clear that there are lots of parasite, especially eggs, so we would find a lot of different sequences there, wouldn't we?
You would have a plethora of sequences.
Someday when it's very cheap,
yeah.
you can take some soil and sequence it for $100.
Right. That's right.
It would be very interesting to go to different places and do that.
Oh, no question. But in the tropics, particularly where sanitation is limited in some places, many places, I should say, this parasite is extremely prevalent. To gauge the sanitation is very simple. All you have to do is line the kids up from school and take a stool sample and look for Trichuris. If it's there, you know that they're being exposed on a daily basis. Right is wow. Wow is right. Wow is right.
What does it do to the kids?
Well, I just told you, if you have a few, nothing. If you have many, you could have an anemic situation.
Anemia.
You could have diarrheal disease. It could turn into dysentery.
It says they have also some inflammatory colitis type diseases.
That's right. That's right.
Because you get an immune response, so you get
That's
inflammation.
correct. But eventually, if you were to, let's say, leave that zone and never go back, your parasites would be gone in about 5 years.
If you don't keep feeding the infection, right?
That's right. That's right. Now, this is the intriguing part. Now, of course, you'd want to intervene before this.
How do you do that?
Well, there are drugs, many.
So you would have some sense that the kids were infected, dysentery or anemia.
And then you'd look under a microscope and do a stool exam. It's called a stool examination.
Right.
And you'd find these eggs, and they're very characteristic. They look like little footballs. And they have a little covering on both ends, and they're called an operculum.
Yes, show here in this diagram, right? Yeah.
And the worm actually gets out by dissolving this operculum, and then out it goes. The little cap pops off, and out goes the worm. And that little cap, by the way, is the point of attachment from E. coli onto the columnar cells. So it's right near where it has to go. Okay. So the cure for this, all right? Not the prevention, but the cure is to give mebendazole.
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 Or mebendazole or albendazole.
That's right. Either one interferes with microtubules, but only in invertebrates. Okay. So tubulin is the target for this drug. And invertebrate tubulin, not vertebrate tubulin. And it works beautifully.
Until you get recontaminated.
Yeah, the problem is, of course, that you just get re-exposed.
Do you get resistance to these drugs?
You mean in the worms themselves?
Yes.
Yes, you do. And in fact, we've been using it so long, even though their reproductive rate is so low, but remember, they produce 5,000 eggs a day.
Sure.
Some of them are bound to be resistant to every drug, right? Just by random mutation. I don't know of any particular zone on Earth that's particularly prone to having a resistant set of organisms to mebendazole, though. I don't know that. And so the WHO and the Gates Foundation and several other initiatives have, on Millennium Development Goals, have included mebendazole into a cocktail of antiparasitic drugs to be given at the community level to try to reduce the worm burden, as they would call it, so that the nutritional status of the community goes up and the health of the kids get higher, the growth rate resumes, and they can stay in school longer. That's the whole basis for that program. There are no vaccines. None.
Do we need a vaccine?
Well, if I had a vaccine against these parasites and it wasn't a particularly difficult vaccine to give, I might consider it.
Could you eradicate this parasite?
Not since the worms live for 10 years in the soil as eggs. It's impossible, I think. The only solution is to move away from that zone.
But it's quite prevalent, so that would be hard to do.
But there's another side to this issue that we're going to talk about now, and that is What good is this parasite? Is this parasite good at all? Or are any parasites good?
Are there, from all the ones we've talked about, are any of them good for us?
Well, Vince, let's put it this way. We evolved with these parasites. Okay, we did 200,000 years ago to the present. Prior to that, as other hominid species. You can find the eggs in coprolites, as you mentioned. I'm certain this was there prior to the advent of Homo sapiens sapiens. What does that tell you? It tells you that the immune system that we have has been selected for fighting off these kinds of diseases. All right? Now, what happens when you take away the target for, let's say, a tank or a machine gun or a cannon? How do you stay sharp? The actual target is the enemy. But we're not fighting all the time, are we? So we train, we're prepared for the enemy, so we take target practice.
Right.
Target practice makes perfect. How does our immune system target practice when there's no target? A difficult question to answer, but I can tell you what the outcome is here. Apparently there's a subset of people who have an antigen or two on the surface of their gut cells and inside as well, but mostly on the columnar epithelium, that resembles an antigen that's a target for Trichuris. And the immune system, instead of firing away at only the worms, starts to fire away at our own tissue. And they create a condition called Crohn's disease. Crohn's disease is widely known, and there are a lot of people that suffer from it. It's not the same as irritable bowel syndrome. It's not the same as wheat gluten sensitivities and all these other food allergies that are associated with malabsorption syndrome. But Crohn's disease is a real nasty disease. It's an autoimmune disease where the immune system attacks the gut tract, the large intestine, and eventually causes the tissue to become friable, to start bleeding. You get something that looks like ulcerative colitis as a result. And the surgeons have to resection that portion of the gut and eliminate it. And essentially, the patient destroys their entire large intestine over their lifespan. By simple chance and good epidemiologic studies, it's been known for many, many years that populations that are heavily infected with Trichuris have no Crohn's disease to speak of. None. And you'd say...
Wait, you just said that Crohn's is precipitated by Trichuris.
I didn't say that. No, I didn't say that.
I thought you said the antigen...
There's a lookalike antigen.
Between Trichuris and the colon.
That's right, but these patients that have Crohn's disease have never encountered Trichuris, ever in their lifetime. Their gut tract apparently resembles Trichuris in some way. And I'll tell you how we know that, because of this epidemiologic study that was done, and lots of others too that confirmed it, that said the moment you take these populations and you allow them to live in places where Trichuris does not exist, or hookworm in this case too, the rate of Crohn's disease starts to become apparent in these people, and the rate of food allergies starts to go up, and all kinds of other skin allergies go up in populations that had none of that before, but of course they were infected with intestinal parasites. Someone got the bright idea of trying to infect a Crohn's patient with Trichuris.
I was just going to ask you, what happens if you do that?
Well, surprise, surprise.
Does
Parasites.
It get worse?
No, it gets better. Crohn's disease goes into remission. The immune system switches their attention to the real targets, which are the parasites themselves, and they start to attack them. And in 3 to 5 years, of course, they kick them out. Which means, of course, that those poor people, even though they had a respite from their parasitism, or from their Crohn's disease, rather, by having a controlled parasitic infection, the moment they kick their parasites out with this very strong immune response that they've got the capacity for generating, they go right back to attacking their gut cells again.
Why don't we give them a protein instead of the whole parasite?
Let's make a molecular vaccine for this thing that keeps the immune system busy on the proper target at least, and get it out of harm's way. and leave those poor people's gut tracts alone. So that's
or give immunosuppressants to
well,
the
they've tried that. You know what? They've tried that, and they get into big trouble there because then they start becoming susceptible to other infections when you do that. Okay, so Trichuris has played a role in the treatment of Crohn's disease, and it's now approved for IRB use. And the parasite of choice turns out to be sheep. Trichuris ovis.
To give people with Crohn's.
Yeah, or Trichuris suis. Yeah. Remember when I said that this parasite doesn't really cross species? Okay.
Right.
It's not entirely true. They'll produce a sterile infection.
What does that mean?
Or very low infection. It means that the parasites will mature inside the gut tract of the patient, but they won't mate and produce fertile offspring.
Okay.
So you can infect them with these parasites. Another approach would be, of course, to collect adults from sheep, okay, and only give the male parasites. The male parasites in this case don't live as long as the females.
You can ingest an adult?
Yeah, you can do that. If you actually inserted them down into the large intestine, they will take up residence.
You put them in via the rectum.
You could do it that way, but mostly it's done by endoscope.
Oh, that's terrible. Put a worm through a tube into your colon?
Well,
I don't think you could reach the colon.
Maybe. I'll withdraw this scenario.
I think that's too far.
Yeah, I'm not gonna stick my neck out on that one. No pun intended. I would presume the other way is the proper way for that to go too. Yeah, I think that through the rectum, through an endoscope, that you'd have a proctoscope. You could actually insert them this way. So that would be another approach to this. And once you establish those infections, the Crohn's disease ablates, it goes away. And it doesn't come back until the parasites are kicked out. So I've heard presentations on this, and in the beginning, everyone thought this was heresy.
Yeah.
But the more you think about it, the more logical it seems, because in our society, we don't have these parasites. We've eliminated them by sanitation. And up springs another disease as the result of that.
Well, in areas where there is a lot of trichuras, how's Crohn's disease? Is it down? Yeah.
0. So that's the epidemiologic surveys in many places throughout East and West Africa and Asia too have confirmed the fact that Crohn's disease does not exist in populations that are heavily infected with these intestinal parasites.
So what you're telling me is that we evolved with this parasite. It keeps things in check. We have symptoms, but if you get rid of it, then our immune system is taking target practice on ourselves.
That's what I'm saying.
Get autoimmune disease.
That's the overarching principle here, and that's why a little bit of dirt is good for you and a whole lot is not.
Yeah, but you don't want a lot of these worms.
No, of course
then you
not.
have the problems.
I mean, it's a trade-off that neither way.
So Harold Brown's one worm was probably good for his Crohn's disease.
Nah, he didn't have Crohn's disease.
That's because he had one worm.
Well, you wouldn't have ablated Crohn's disease with one worm anyway.
How do you know, Dickson?
Because they did dose response curves on these people.
I'm just being devil's advocate.
About 100 worms, 200 worms is about right for an adult.
I don't think we should give people worms to treat Crohn's. I think we should find a
No,
molecule,
I agree with you,
right?
but in the meantime, we don't even know what the molecular target for the parasite is from the immune system.
Yeah,
so
well, you have to sort that out. And
with what? Wait a minute. That's true. I would agree with you.
You want to know what.
So how do you do that?
I mean, I know that in Crohn's, the innate sensors have been implicated. So the membrane receptors that sense pathogens are overactivated here, and that's part of the problem. So if we can calm that down, maybe you can regulate Crohn's.
Maybe.
But I don't like the idea of giving an infection to...
No one does, including the Crohn's patients, but they feel so much better afterwards that they rave about it.
Because I know, something could go wrong. Maybe someone...
Well, that's what the original thought was. Maybe you could introduce a virus this way, Vince.
Well, we can use viruses for gene therapy as long as we disable them, right?
Yeah, that's right.
Or gene therapy or some other... You can imagine delivering something here by a gut virus.
That's
right. Sure, but I don't want the parasite to grow because then you can have unexpected consequences, right?
This is all true, except that we have so many years of experience with little kids infected with Trichuris that there's very little danger of exceeding the limits with this particular parasite.
Aren't there some other parasites that are involved?
Yeah. Hookworm is another one that they've used, and it actually does work as well.
There was a statement here that caught my eye.
What's that?
And let me see if I can find it now. Yes. There's evidence that, in addition to the problems we've talked about, anemia, diarrhea, dysentery, prolapsed rectum,
malabsorption,
there can be cognitive and intellectual deficits.
This is true, but that's particularly true for hookworm, as we will discuss.
So that's another reason not to use this as a therapeutic.
Nah, you know what? If you had Crohn's disease, you wouldn't say that.
I know.
It's a trade-off.
I understand.
It's a trade-off. In fact, one of the authors of our book, Peter Hotez, is a world expert on the effects of intestinal impairments on learning in small children, because he's a pediatrician by training. So that's his contribution to this, is that he's very concerned about early cognitive learning in patients that have heavy worm burdens. So let's summarize Trichuris, okay, and compare and contrast it to Anaerobius. They both produce eggs, but Trichuris eggs take a week to embryonate in the soil, and Trichuris eggs will embryonate right on the perianal region, 6 hours versus 1 week. The adults of pinworm, when they lay their eggs, they die. Trichuris adults live 3 to 5 years.
Mm-hmm.
Adults of pinworms do nothing except elicit an allergic response to their proteins when they disintegrate on the perianal region, which causes secondary infection due to scratching at night when the kids are asleep. Trichuris adults, enough of them, can cause anemia, diarrheal disease, dysentery, malabsorption syndrome, and cognitive learning defect. Serious parasite.
Yeah, it's a serious problem.
Serious problem with hundreds of millions to 1 billion people infected with this. So 1/7 of the world has this parasite. That's incredible, Vince. That's incredible. It tells you how little we've advanced in terms of our origins, in terms of our ability to live apart from the things that can do us great harm.
Well, for the underdeveloped part of the world, that's a problem for many different microbes.
This is one of the defining features of the undeveloped world. That's how you know that you're in the underdeveloped world.
Is that right?
Or the less developed world.
None of the other parasites we've talked about so far are defining features of the underdeveloped
world?
No,
we've talked about Giardia.
Malaria, no?
Oh, yeah.
Giardia? Well, Giardia, the campers.
And in fact, you might argue that the reason why they are less developed is because they're so busy fighting off these diseases that they haven't got time to develop.
I would argue that there's a whole collection of
there's a constellation of reasons, but this sucks off a lot of economy from a less developed country because people don't work as hard because they can't when they're heavily infected with these parasites.
Okay.
And the infant mortality rates are high and the adult death rates are high.
But the pinworm is not a defining third world because obviously we talked
it's
about
ubiquitous.
That. It's everywhere.
It's everywhere. It's everywhere.
Well, I think we should do some email now.
Well, that was a refreshing...
That's fine. I think it's good to cover things comprehensively.
You bet. And of course we didn't, because we've left lots of gaps of knowledge here.
Well, we will return.
We don't have that knowledge, Vince. That's the problem. So I was going to say to you, in response to your, well, we have to know more about this and this and this. I totally agree with you. But at the same time, you need funding for this. In order to know the answers to those questions, you have to get funded. That's the name of this game right now. And there's hardly any funds at all available for this kind of parasite.
Well, if you're good, you'll get funding. That's the bottom line.
Too bad.
But you have to be good, and...
You have to be better than good.
Let's just do a very quick PubMed search for Trichuris and see what the most recent paper is. I'll
bet it's a survey paper.
Oh, yeah. It's from December 2010, Epidemiology of Intestinal Helminth Parasites in Stray Dogs from Markets in Nigeria. Wow, they must eat. The dogs are running around the markets, I guess?
Oh, yeah. Well, they've got their own species of Trichuris, that's true.
Mexico, a survey.
See, surveys.
Sierra Leone, a survey.
Try to see if you can find
here's one. IL-22 positive, CD4 positive T cells are associated with therapeutic Trichuris infection in an ulcerative colitis patient. So mechanistic.
Aha! What about the E. coli association? See if you can find that one.
Let's do Trichuris and E. coli. Got 56 returns here.
That's good. It's a very recent paper.
Recent paper. So the most recent one is Science 2010.
Which says,
here we go. Exploitation of the intestinal microflora by the parasitic nematode Trichuris murus.
We should list this one on the.
I'll put them in the show notes.
Put it on the show notes. That's the paper, and it's a riveting study.
Nice.
Now remember, I did my studies at Notre Dame on germ-free animals and Trichinella.
Right.
Is there an attachment mechanism there for E. coli? And the answer is
sure.
There can't be, because the worm lives in the small intestine, not the large
Well,
intestine.
are there bacteria there? No, it's sterile.
Largely.
I think this
E. coli lives as an anaerobe.
idea that pathogens require flora is probably more prevalent than we think for the ones that...
It's a community effort.
Let me just give you a little clue. Polio replicates in the colon as well as the small intestine, so I bet there's a role there. All right, let's read a couple of emails, Dickson.
Okie doke.
The first one is from Jim, our friend from Virginia, who says, for the TWiP file, it sends us a link to a Science Daily article called Getting Rid of Cattle Fever Ticks. Scientists at the USDA have developed 2 strategies to ward off cattle fever ticks that are crossing the border from Mexico into the U.S.
Without passports, I might add.
These ticks transmit bovine babesiosis, or Texas cattle fever.
The first association of an insect or an arthropod-borne disease, by the way, that was discovered by Theobald Smith.
Really?
Back in the 1800s.
Yes, that's right. We've talked about that before.
Yep.
So they have a bait station where they lure the deer, and then they get automatically insecticide applied to them.
So it's a cattle tick, but it also affects deer.
Yeah. New interventions too. So I guess it's brought over because cattle are not wandering around, right, Dickson?
They are wandering. No, they're wild.
They're wild cattle?
Oh, they're all over the place.
White-tailed. Okay, so the increased spread of infestation is due in part to the increased populations of white-tailed deer and other hoofed animals along the Texas-Mexican border.
Right.
So here they're showing how they can get the deer to be de-loused. No, not de-loused, de-ticked.
Right.
Cool. So we'll put that link in the show notes. Thank you, Jim. The next one's from Mark. Dear Vince and Dickson, wonderful job on both podcasts. I have just finished listening to the Cryptosporidium episode.
Cool.
I love all topics regarding the Apicomplexa.
Excellent.
And unless I'm mistaken, there is a fascinating aspect of this phylum which I have not heard discussed on your show. This is the apicoplast. As I am sure Dickson is aware, sometime in the early 1990s, a degenerate plastid was discovered in Plasmodium and later discovered in most apicomplexans. Interestingly, it was also noted that Cryptosporidium lacked this organelle, and present evidence indicates that Gregorines, another type of apicomplexan, also lack a plastid. The presence of this plastid is interesting for many reasons. Primarily, this organelle may provide drug targets that would not interfere with host biology due to the cyanobacterial origin of plastid. However, what I find most fascinating is what this implies from an evolutionary perspective, that modern-day bloody parasites like Plasmodium have an algal/photosynthetic ancestry.
True.
Talk about wild evolution.
Yeah.
This has been exemplified by a discovery in 2008 of Chromera velia, a photosynthetic alga that is close phylogenetically to the Apicomplexa.
Wow.
Evolutionary implications of this plastid are indeed fascinating from both the perspective of horizontal gene transfer as well as the evolution of the relationship between these parasites and their hosts. These relationships are ancient indeed. These discussions tend to stray from topics regarding virulence. However, I feel your listeners would enjoy hearing something about the evolutionary history which developed these truly chimeric yet single-celled critters. Keep up the good work. So
nice.
I think we will have to get some expert on this.
Oh, yeah.
To join us in a future episode.
Make a note.
Because this is pretty neat.
Absolutely.
Thank you, Mark. Carolyn writes, Dear Vincent and Dick, I'm really enjoying your podcasts on parasite. I took special note of the one you did on Cryptosporidium. Second one.
Okay.
I'm a 52-year-old beginning backpacker. In doing the research on water purification, I found that Giardia and Crypto were the two that came up the most. Since I would prefer not to use a mechanical filter and don't mind the taste of chlorine, I want to use a Myox system, a superchlorination system using table salt and a battery. The instructions say that it kills Giardia within 30 minutes, but takes 4 hours to kill Crypto. In listening to the podcast, I found that every time it looked like some questions might get answered, the point of the topic shifted to something else. I'm sorry. It's my fault, right?
No, not at all.
Here are the conflicts I have. When I hiked Pikes Peak, the bar camp personnel said they only knew about protecting against Giardia. Is crypto not something to really worry about, or is it really everywhere?
Crypto is mostly a cattle disease, and so that, in terms of Pikes Peak or hiking in the meadowlands, the upland meadows, in many cases you can find free-range cattle in those areas, and you would have to worry about it. But in other areas, like national parks, for instance, where they're not allowed to have cattle, at least they shouldn't have cattle in the national parks. Not in the national forests, but in the national parks. Then you might not have to worry about crypto. But I would recommend boiling the water, but if you're at a certain altitude, the temperature of the water boils at a lower temperature, and therefore you're also stuck on that one. Gee, I don't know why you're not favoring those porcelain filters. They're pretty good, and I would want to look at that one again.
She says, you mentioned immunity. If a person is immune, does that mean they won't have symptoms? Symptoms are less. I drank of a lot of creeks and even ditches when I was a kid. Am I immune?
Good question, and it depends on whether you developed infections or not from those behaviors. We wouldn't recommend doing that now either, by the way.
So if you were infected
with crypto, for instance,
you'd never get it again.
Well, the assumption is that there is a long-lived immunity, and we're not sure what the mechanism is, so we can't honestly say whether it fades over time or not. Or it may actually be re-stimulated by exposure again and again in terms of that. With Giardia, you can catch it again and again. So that's one of those infections that, although you do develop immunity, it can come back. Maybe at a lower rate, but it still does come back.
Fewer symptoms or less severe symptoms.
Right, and cattle behave the same way. A newborn cow, a calf that's a weanling and then no longer suckling on mother, gets infected with this and develops a projectile diarrhea, basically, and throws out millions of oocysts from crypto. And then as it gets older, if it survives this stage, of course, it gets more and more refractory to infection, and finally, as an adult cow, it's hard to find it.
I saw a crypto outbreak somewhere just recently.
It's all over the place.
All the time. Water supply.
All over the place. Foodborne, water supply, all over the place.
So she says, you said chlorine won't kill it. Crypto.
That's right.
That's true?
Yeah.
Will Myox solution kill it in 4 hours, or are the instructions wrong?
Don't know the answer. Sorry, I can't tell you.
Myox is, I guess, so they take table salt and crack it to chlorine. That's what we do in our swimming pool.
Right.
So you don't have to add actual chlorine.
I don't know the answer, to be honest with you, but I would trust a mechanical filter more than I would
She says the Army uses Myox.
Okay.
She said, I really don't want to get sick, but I can't lug around gallons of water every time. I mean, so Dickson, your solution is to use a mechanical filter. And
I use one, and it's fairly lightweight. I go trout fishing all over the place, and I do get thirsty in the daytime, of course, when I run out of my bottled water. And, I pump the water out of the river, and I drink that. That's my solution, but everybody has their own preferences.
I'm looking at the Myox site right now to see what they say. Well, you say that they say it kills...
But 4 hours, that's a long time. If you start treatment of your water supply as you make camp, for instance, in the evening, 4 hours later it's dark. So it's a long time to wait for pure water, I would say. Whereas with the pump, you can have it right away. But again, it's a personal preference.
I'm looking at the CDC page on crypto, prevention, how to stay healthy. Let's see. Recreational water venues, water-related hygiene. They're not saying anything about treating it. Crypto and water filters.
There are lots of good ones out there.
So they talk about water filters.
Yep.
For crypto and not any kind of treatment at the CDC.
Right.
Which is consistent with what you feel, right?
Well, yeah. I mean, I'm a big fan of the porcelain filters that are available nowadays. They're pretty good.
Okay. So we don't know about...
In fact, some of the recent iterations of that have resulted in this straw that they can give to little kids living in less developed countries. And they can sip their water right through the straw, and there's a filter in it. So they don't have to drink directly out of the river. They can drink through their straw. And eventually it does clog up, but it takes a long time for that to happen.
So Myox, you don't know, but you would filter it to be safe.
I would. That's my bottom line. No pun intended.
Yeah. Okay. So this is from Sam. Okay, Sam has a new suggestion for a catch at the end of our shows. We say another TWiP is parasitic. He said, how about this? Another TWiP is hosted. Another TWiP is vectored.
Arg.
Another TWiP is spiral.
Oh, my goodness.
All right, well.
But thank you for trying.
Thank you for trying. Jesper,
it brought a smile to my face.
Jesper from Sweden writes, All in the Mind is a great radio show from Radio National Australia, mainly about things pertaining to the brain. They did a show about parasites and their effect on populations. You'll find it here, including a transcript.
Nice.
One comment references Radiolab, which I must assume is the one where Professor Despommier was featured. Probably Dickson, right?
Maybe.
You're so cautious. Here it is. All in the Mind. We have ways of making you think. Parasites on the brain. Half of the living things on the planet are parasites, and they've developed uncanny ways of making us do what they want. A spooky encounter with suicidal crickets, mad rats, and zombie ants. The parasites are here to take over our brain. One ecologist argues that they could even be changing our personality at the civilizational scale. Okay, well, you know, these programs tend to be very spectacular. They try and make huge claims.
Yeah.
Otherwise, people don't listen. But here on TWiP, people listen to TWiP, and we don't do that.
We try not to, at least. That's true.
I don't think you have to be sensational to have an audience, do you,
These
Dickson?
parasites are sensational, Vince. Believe me.
Thank you, Jesper. Felix writes, Hi, professors. I don't think in your program on crypto that you actually said by what mechanism it causes diarrhea, whereas you have given similar explanations for other organisms.
Yeah, that's because we don't know.
You don't know?
We don't have a clue. This parasite actually creates a special landing site. That's the best way to put this.
Mm-hmm.
It actually forces the microvilli to fuse and grow up around the parasite and encompass it in a membrane-bound organelle that it creates for itself. The bottom of which, on the surface of the columnar cells, develops into what looks like a feeding mechanism for the parasite, where it taps into the actual columnar cell itself. And maybe in doing that, it induces a hypersecretion of the columnar cells, and maybe that's what the diarrheal disease is from, but that still doesn't tell you what does it. It just says maybe that's where it occurs, but it doesn't give you the mechanism by which the hypersecretion actually exists. And remember, I said in patients suffering from AIDS that caught cryptosporidium before we had the triple therapy, they could produce up to 10 liters of very watery stool every day. That's an incredible number of bowel movements. And you have to replace all of that fluid and the salts at the same time. And they still don't get better. So, and we've tried everything to restore their immune responses to normal levels. Tried every drug, didn't work. The only thing that did work is to restore their immune system and let them take over.
Interesting. But you have to give them fluids, though, otherwise they'll die.
Yes. Many of them died from it because we didn't have the triple therapy available.
All right, our last one is from Attila. Dear TWippers and Twivers, after listening to all TWiPs, although I will listen to TWiP 19 in some minutes, I have noticed many readers send suggestions of parasites that manipulate hosts' behavior, such as cordyceps fungus. So I wanted to make my contribution too and add to the discussion of modifying mosquitoes to bite less and decrease malaria transmission.
I would love that.
The problem I see in this approach is that the Plasmodium already does it. Plasmodium is able to make the infected mosquito bite more frequently, thus increasing its transmission. He has He has a paper here.
Yeah, I think that's controversial literature, but what I would like to see is a follow-up on this, and I agree with you. If we could discuss this in person with each other, we'd probably both agree on this, that most mosquitoes bite once and then lay their eggs and die. All right? Why do some of them, like 1% of them or less, bite twice? Because if they only bit once, they wouldn't transmit the malaria parasite because they couldn't have picked it up to begin with, right?
Right.
So my goal would be to genetically modify all mosquitoes. You can have your blood meal, but you can only have one. So there's something in your blood that shuts them down that you've put into it, like with a tablet that you take or something like this, that shuts off the hormonal regulation of egg production.
So you want to do this to all mosquitoes?
Every one of them.
Even the salt marsh mosquitoes?
All of them.
They don't transmit anything, do they?
Oh, yes, they do.
What do they transmit?
Encephalitis virus, Vince. Eastern equine encephalitis, the salt marsh mosquito, is the best.
I asked the Key West people, and they say in the Keys, those salt marsh mosquitoes don't seem to have anything.
In Jersey.
In Jersey, they do.
And remember, this is a vertical transmission with viruses. Not so with protozoans.
Well, he gives a paper called Malaria as a Manipulator, 2005.
Who produced that paper, if I may ask?
Coella, J.C. Coella. We'll put a link to it. And then he says also it is not only the mosquito that Plasmodium can modify, it makes infected people more attractive to mosquitoes. And he has a paper from 2005, Malaria Infection Increases Attractiveness of Humans to Mosquitoes in PLOS Biology. As a regular TWiP listener and a big fan, I like the differences between the two podcasts. Professor Despommier's impeccable narration, I liked this letter, makes parasitism really interesting. I had a terrible impression of parasitism in college. It is a big topic of biology in Brazil, since there were many parasitic diseases not so long ago, and some parts of the country still have it. And the lectures were all focused on long life cycles and lots of different names. Only after reading the histories behind the diseases, the patients, and the discoveries, as well as learning the host-parasite interactions, I started to like this topic. Carl Zimmer's texts had a big role in this change, and I'm very fortunate to hear someone with so much experience and personal history as Dick Despommier in TWiP, especially now that he is a busy pop star and best-selling writer. Hey, you're a pop star.
Well, I'm a pop. I have 2 kids.
You're a pop star. That's why you're busy.
Pop star.
Long and prosperous success to TWiP, Attila. P.S. I have great expectations for the guests you may invite, given the quality of TWiP and the guests of TWiV.
Not a problem here.
Thank you. Yeah, we will come to it.
We will dazzle you.
We don't know when yet, but we get to get through all these basic discussions.
That's where we're heading here.
And then eventually we will...
And then we'll start topics in parasitism and for research. Research topics in parasitism. What's the latest and the greatest?
Right. Well, that will do it for another TWiP. You can get your TWiP fix many ways on iTunes at the Zune Marketplace. You can go to microbeworld.org/TWiP. You can play it there. You can download the episodes. You can look at our show notes. You can also listen on your iPhone or iPad. There's an app at microbeworld.org where you can do that on your Apples and on your iPhone and iPad, but also now on Android devices. It is available.
Nice.
And as always, if you go to iTunes, leave a review to keep us on the front page of the Medicine Podcast section. As always, send us your questions and comments to TWiP at twiv.tv. Dickson Despommier, thank you so much for once again explicating parasites.
It's my pleasure, Vince. You know that.
And it is my pleasure to explore them with you. I am honored to be able to do so. Thank you very much.
Feelings mutual.
I hope that you are always interested in educating the world about parasites.
Of course.
And who else better to do it?
It's my passion.
You are at Trachinella.org.
I am.
As well as verticalfarm.com and medicalecology.org.
Yep.
Right? Those are the 3 websites. And I am at virology.ws. Of course, I am Vincent Racaniello.
You are.
And
he's always been Vincent Racaniello.
I've always been since birth and will always be.
That's right.
You have been listening to This Week in Parasitism. Thanks for joining us. We'll be back next year. Another TWiP is parasitic.